of the IPC. Good afternoon and welcome to the 10th session of the IPC ECHO. It's a monthly series going up to 16 sessions. You are welcome. Today's session is taking us back to the basics. We're learning the basic microbiology for about 45 minutes. We do a crisp presentation, a question and answer session, and we'll have 5 minutes to share our feedbacks. Introduce the host team. We have Lydia, we have Romina, Nicholas. And before I introduce our key speakers, I would like to remind us we need to create a conducive learning environment whereby we interact mainly through the chat box. Raise your questions, comments on the chat box. Kindly keep muted so that we can all be attentive to the presenters. Before I go further, Lydia, I believe all the systems are set. I would want to introduce the first speaker, if you can allow me.
Yes, Mark, can you go ahead?
Thank you so much. Our first speaker is a professor I've known for almost 10 years. She's a Professor Revadi Gunturu, who's a current head of microbiology in the Department of Pathology at Khan University, who joined there in 2006 and established crucial sections of diagnostic microbiology and establishing the teaching programs in the clinical microbiology in Khan University. Prior to joining this position, she held positions of head of department of laboratory medicine at Kenyatta National Hospital and Associate College of Health Sciences, University of Nairobi. She received a MD in clinical microbiology in 1984 from Osman University, India, and she was the faculty member of clinical microbiology at Maulana Azad Medical College and University College of Medical Sciences of Delhi, University in New Delhi, India, for 10 years before moving to Kenya. She's a visiting scientist at KEMRI, where she has active collaborations up to date. And she supervised numerous postgraduate dissertations projects throughout her career. And so far, she has published 90 papers in peer-reviewed journals. She's a member of several national and international professional bodies, about 7 of them, and she's including KMA, GAP, among others. Sorry. She's currently the chair of IPNET Kenya, a multidisciplinary society that promotes education and advocacy for IPC and control IPC in the region. She's a member of national advisory committees, such as the National Committee on Infection Prevention and Control, National Antimicrobial Stewardship Committee, and the National Technical Working Group for AMR Surveillance of the Ministry of Health in the Republic of Kenya. Welcome, Professor Revadi.
Thank you so much, Bogori, for that very generous introduction. Hello, everyone. It's my pleasure this afternoon to share a little bit of basic microbiology stuff with all of you. And thank you, organizers, Lydia and the team, for giving me this lovely opportunity. I think I'll keep my video closed because of bandwidth issues. Let me share my screen. Yes. I have no conflicts regarding this presentation, and the source for this presentation is a lot of online resources, free websites, and special thanks to Sarak Jumali. This is my hospital, Aga Khan University Hospital, Nairobi. A lot of you are familiar with this. This is my laboratory where the microbiologists are running bacteriology tests, as well as molecular diagnostic tests. This is our core laboratory, just to share a few glimpses of what I do, where I work. And this is microbiology. What is the role of microbiology in all the operations we do in the healthcare setting? We have microbiology laboratory, which connects directly or indirectly to all healthcare systems within a healthcare facility or within an administrative structure. Pharmacy, you can see on this slide, the red arrows are going bidirectional. Pharmacy, the arrow is up and down. Medicines are dispensed and sold, that is antibiotics, especially we are interested in when it is microbiology, and infectious disease diagnosis, and how much antibiotics are used, how much are sold, and it connects directly to microbiology because microbiology is reporting antibiotic susceptibility tests to the doctors. And the other one is clinical areas, all the wards, medicine, surgery, pediatrics, ops, gynae, everything. And what is the outcome? What is the diagnosis? How are the patients getting treated? That and reports are going from microbiology lab to the clinical areas. So this arrow is also going up and down. Now administrative, the third square is administrative, where how much money is spent on each patient admitted, how much money is spent on antibiotics, how much money is spent on diagnostics, and how long did this patient stay. All these are administrative issues. They also directly connect to microbiology. This is a summary of microbiology susceptibility results, which periodically Aga Khan University Hospital prints every year, summary of microbiology spectrum and susceptibility. This is just an example. And this is how microbiology laboratory in my hospital, in my laboratory, microbiology, they circulate summaries of antimicrobial susceptibility data. This particular table is showing organisms. It is also showing antibiotics on the several columns. Total number of organisms. Let me take the pointer. Yeah. These are the organisms, and year, cumulative, these are the numbers of each organism. E. coli in 2012 was 1122. These are the antibiotics. All these are antibiotics, and this is percentage of susceptibility. Ampicillin was only 20% susceptible. It means 80% was resistant. Meropenem was 100% susceptible, means 0% resistance. Whereas Septrin was only 24% susceptible. So this is summary cumulative susceptibility data, which microbiology lab circulates. So that is another cardinal functions of microbiology laboratory. Microbiology lab also dictates the quality of samples and is continuously engaging all the paramedics, medics, pre-medics, and clinical staff about the sample collection, which is the correct sample, when should it be collected, how should it be transported, and then within the lab, they have what are called rejection criteria. If any sample is poor quality, the lab rejects. So all these are functions of microbiology. This is just an example of what is the right sample. This is a wound. If the wound is like this, dry, and there is no discharge, no collection, swab is a correct sample. Here, there is a lot of necrosis. Curettage, a tissue, is the right sample. This is a swollen lesion. There is a collection of pus. The clinician can see, he can examine. Aspiration is the correct sample. Put a needle, clean the superficial area, put a sterile needle and aspirate the sample. So this is going to be the correct sample to get the correct diagnosis. That is, lab is able to isolate the correct bug and is able to give you the correct susceptibility results. So this is also the docket of microbiology laboratory. What is the correct sample selection, collection, and transportation? Then specimen collection management is another area. How is the specimen managed before it reaches, after it reaches? And then we come to basic microbiology. Microorganisms are living things. which individually very small to be seen by naked eye. That is why we say micro. Micro is too small to be seen. And then we divide them into bacteria, fungi, protozoa, microscopic algae, viruses, and various parasitic worms. All these are the various groups of microorganisms. Microbiology is the study of microorganisms. And this is also foundation for modern biotechnology. Among many of the specialized fields of microbiology, you have all these lists. A lot of you are aware of this. Just to recapitulate: virology, mycology, bacteriology, immunology, microbial ecology, biotechnology, environmental microbiology, food microbiology, forensic microbiology, molecular microbiology. Now, these are only a few. I can also add soil microbiology, marine microbiology, and also now what is frontier of research, the microbiology of the deepest areas of earth. That is when a miner is digging deeper and deeper into the depth of the earth, there are microorganisms. Even they are giving us a lot of challenges. They are also giving us lot of solutions to health problems. There are microorganisms which can survive at 90 degrees Celsius in the depth of the earth, in the soil. In the depth of the ocean, there are fascinating microorganisms. So microbiology is a very, very exciting field, let me tell you. Two main themes involved in microbiology. Basic microbiology talks about the cellular processes, the anatomy of the microbes and their physiology, and how they reproduce, how they resist environmental factors, certain things. That is basic microbiology. Applied microbiology is concerning how this knowledge is applied for human benefit, like agriculture, industry, of course, health. That is our main interest here. These are all themes in microbiology, basic and applied. Basic is by organism. You have bacteriology, mycology, virology, parasitology. We have seen all this. By process, microbial metabolism, genetics. By disease, immunology, epidemiology, and etiology. This is what basically in the medical field we are studying: immunology, epidemiology, and etiology. And disease related: infection, infection control, and chemotherapy. Antibiotics, antimicrobial resistance, mechanisms of antibiotic action, mechanisms of antibiotic resistance. So this is a disease related theme. Environmentally related: environmental microbiology and industrial microbiology. Food industry, breweries, making of beer, making of cheese, preserving milk, preserving butter, preserving cream, all those are food technology or food microbiology, which is a very big field. And these are the microorganisms which are too small. They rapidly grow. They have a habitat. They live in population. They never live alone. They live in colonies or communities. Communities are either swimming freely or attached to a surface. Now, when these communities of organisms are attached, they are called biofilm. They interact within themselves. They sometimes counteract each other. They fight with each other. Sometimes they live together, very, very friendly, augmenting each other. Now, what are the microbes in our lives? Some of them are pathogenic. They cause disease. So we are specially interested in them because we want to keep our health safe. Some of them decompose organic waste, and they're very, very important. If they don't do that work, the waste material on the earth will completely drown us. But the decomposing organisms are helping us by disposing of, digesting the waste. Products through photosynthesis, purple sulfur bacteria which fix carbon dioxide. Now we have what are called nitrogen fixing bacteria, which are very important in agriculture. They are the ones which make our beans very rich in protein, which make our other produce very, very nutritious. Producing fermented food, vinegar, cheese, bread, and also our popular beer, wine, all those things. Producing products used in manufacturing, cellulase, and a lot of products which are manufactured, they are all products of bacterial action. Okay. These are the main branches quickly. Food technology, lactococci, which are fermenting milk, producing cheese, and also producing yogurt and butter. Water treatment, there is a lot of bacterial action. Energy, producing methane, bacteria take part and produce methane. And lot of work in science laboratories, and now disease producing organisms. So these are the microbes in our lives. Microbes in agriculture, I won't go into details. Nitrogen fixing, I have already talked about. Rumen microbes helping digest grass and hay in cows and sheep. And a lot of nutrients are cycled by the microbes. So there is a lot of microbes and importance in agriculture. This is the microbes in food. Microorganisms and food, preventing spoilage, assisting in manufacturing of food. Microorganisms and energy, again, natural gas, ethanol, and bioremediation. And microbes and the future is genetic engineering. We are able to manipulate a lot of microbes, and then we have a lot of products for health. For example, the genetically engineered vaccine or the messenger RNA vaccine, which we are using for COVID, is a product of this biotechnology and genetic engineering, as all of you are aware of. Microbes and disease. Now you have a lot of pathogens which are causing disease. E. coli on top, HIV, herpes, ulcer producing, mad cow disease. This is a prion, pox virus, common cold virus, parasite, so on and so forth. There are lots of pathogens. These are all the benefits of microbes: food, environment, prevention of disease, agriculture, energy, wastewater treatment. Just to mention a few. How do we name the organisms? Usually, there is a famous Linnaeus system of scientific nomenclature, and the organisms are named, they are divided into genus, and then there is a specific species or species epithet. And just an example, most of the names are of Latin origin. Escherichia coli. Escherichia is the microbiologist. Coli means lives in the colon or gut. So that is how the names are given. Staphylococcus aureus. Aureus comes from gold color. Staphylococcus means grape clusters. So Staphylococcus aureus, which is living in on the skin, and it has a structure like grape-like clusters. The colonies have golden color, so the name is derived. Most of these are Latin, Greek words, so we find them a bit difficult, but we get used to them. And this is the basic anatomy of bacteria. Prokaryotes and eukaryotes, this is the broad division. And also archaea, that is very, very old, billions of years old organisms, which don't have in their cell structure peptidoglycan. They live in the extreme environments. They include the methanogens, extreme halophiles, and also thermophiles, which I described, which can survive at 90 degrees Celsius in the depth of the earth. And prokaryotes are the ones we are familiar with. Lot of our parasites and a lot of others, they are capable of binary fission, and they utilize organic material, and they also obtain their energy on their own with photosynthesis. So these are all divided as prokaryotes. Protozoa and algae are the typical examples of eukaryotes, which are advanced organisms. And these are the differences. I won't go into these details, which may be a bit more. Fungi. These are filamentous organisms. They are advanced creatures. They have a special chitin in their cell wall. They use organic chemicals for energy production. Molds and mushrooms are typical examples. And yeasts are the other type of fungi. Unicellular fungi are yeasts. Multicellular fungi, which we are showing in this picture, is mucor, and it is rhizopus and aspergillus. These are the examples. This picture is showing rhizopus. Rhizopus has something like roots which are called rhizoids, and it has spores sporangia. This is a typical filamentous fungus. Viruses, very small, cannot be seen even by ordinary microscope. They only have nucleic acid at the core and a cover, a protein cover, and they are the smallest living creatures with some exceptions. So virology is a special science. But there are viroids, organisms without the protein coat, just nucleic acid, and prions, which are just protein molecules. There is no other structure at all, just protein molecule which is able to divide and able to infect animals. These are called prions, and mad cow disease is a typical example of a prion infection. Multicellular advanced parasites are helminths, flatworms, roundworms, and multicellular other parasites. So these are all just to recapitulate the science of pathogens. How do we see microorganisms? There are units of measurements, 1 micrometer and millimeter, nanometer, micrometer. These are all the measurements. And when we can measure organisms with the help of micrometers, which are fixed on the microscopes. And it is possible actually to visualize and measure our parasite eggs, some of the bacteria under the microscope by employing the micrometer. Your human hair, typical human hair is about 100 microns. A red blood cell is around 7 to 8 microns. So these are roughly the natural sizes. If you say WBC, usually it has 10 to 14 microns, and a small lymphocyte may be about 9 to 10 micron. So we have some natural sizes with which we compare. This is typical light microscope quickly, and these are all various types of microscopy. We have stereo microscope, scanning electron microscope, transmission electron microscope, but the ordinary microscopes which we use in the laboratory for routine purpose is called light microscope. And I must mention, between 1857 to 1914, about 50 years of period was called golden age of microbiology. Why it was called so is within this 50 years, a lot of these luminaries who are on this slide, Robert Koch, Joseph Lister, Bejesnik, Paul Ehrlich, and Alexander Fleming, all these stalwarts worked within this 50 years, and they produced tremendous progress in microbiology. Robert Koch, all of you know, you have studied in your school even, about Koch's postulates, what all he discovered, made so many discoveries in bacteriology. And Joseph Lister about the antiseptic technique, first time, and also use of carbolic acid as a disinfectant. He was the first one. And discovery of viruses is ascribed to Martinus Beijnik. And Paul Ehrlich introduced the concept of chemotherapy, actually. And then Alexander Fleming, the discovery of penicillin. And these people, they died, but they always live. As long as the stars and the planets, we count them, we count these luminaries. So this particular time when these people were working, making discoveries, is called golden age of microbiology. And Louis Pasteur, 1822 to 1895, made a lot of discoveries about sterilization, aseptic techniques, making of alcohol, fermentation process, anaerobic organisms, lot of things he described. And he is actually called grandfather of microbiology, Louis Pasteur. And Koch's postulates, I won't go into details, but a lot of these things have changed now, but we still recapitulate these as a very, very fundamental to microbiology. And these were the rules which Koch introduced for pathogenicity and a pathogen. Agent must be present in the case, agent must be isolated, the agent must reproduce the disease in an animal, all these things he introduced. They changed with time, but we still remember them. Preparing smears for staining is a fundamental procedure in microbiology. We will look at it. I'll show you some examples later. And staining makes use of basic dyes and acidic dyes. And there is a chromophore which produces color. And with the help of the color, you stain the smears and then you put them under the microscope. Then you examine the microorganisms. And we have famous stains: simple stain, differential stain, which is gram stain, and acid fast stain. And these are the basic procedures in a microbiology lab. You will always find them. And organisms based on the gram stain, they are divided into gram positive and gram negative organisms. This is one of the flow charts for dividing gram positive organisms. This is the flow chart for gram negative organisms. I don't want to frighten you people with all this. This is for a microbiologist, but simple things. These are special stains. This is example of gram stain. You can see the blue organisms and the pink organisms. And you can see the pink rods, which are gram negative rods. And I'll share with you some more. This is the fundamental structure of the cell wall of gram positive and gram negative bacteria. And this is very, very important because the antibiotic activity, antibiotic resistance, they all are invested in this particular structure of the bacteria, the cell wall anatomy. So this is a bit important. So this is basically the anatomy. This is a grape-like cluster, gram-positive cocci. Cocci means round organisms. These are rod-shaped gram-positive organisms with a flagellum. Flagellum is a tail or a hair. And this is a chain of round organisms, which we call it streptococci. If it is like a grape-like cluster, it is staphylococci. If it is like a chain or a necklace of pearls, it is streptococci. Now, these are microscopy pictures. These are gram-negative cocci. These are gram-positive cocci. These are gram-positive bacilli, which are rod-shaped. These are gram-negative rod-shaped, which are bacilli. And this particular picture is showing you gram-negative bacilli, gram-positive cocci. I think now everybody will really identify. Now, this is at the core of microbiology. For the purpose of most important learning for other professionals who are not pathologists, who are not working in the lab, this is the essence of what should be going on for the purpose of IPC, AMR, and AMS, antimicrobial stewardship. Bacteriology tests, identification of bacteria, and antimicrobial susceptibility testing is at the core. The disease conditions everybody should make himself familiar, any medic, paramedic, is sepsis, either neonatal or adult, fever, lower respiratory tract infection or pneumonia, urinary tract infection and complicated urinary tract infection, meningitis, and enteric fever, or any other enteric infection. So these are the broad categories of infectious bacterial diseases, and these are the top priority WHO pathogens. Critical priority is E. coli, Klebsiella, Pseudomonas, Acinetobacter. High priority is Staphylococcus aureus, Salmonella, Streptococcus pneumoniae, and Streptococcus pyogenes. However, we have several other organisms which we isolate in the routine microbiology laboratory. Then one more example I want to share with you. Any pathogen will have some common virulence factors. This is for Enterobacteriaceae, that is the gram-negative organisms which live normally in the human or any other animal gut. Typical example being Escherichia coli and Proteus. Proteus is always present in the human or animal gut. These are all the typical virulence factors: endotoxins, capsules, antigenic materials, secretion systems, sequestration factors, resistance to the killing activity. These are all the virulence factors, and antimicrobial resistance can also be added as a virulence factor. So this is quickly the anatomy: cocci, bacilli, chains, clusters having flagellum, and one more is a helical bacterium, Helicobacter pylori. Treponema pallidum is a spiral bacterium. So these are special organisms. Vibrio cholerae is another special morphology. It is a comma-shaped organism and it is highly motile with the flagella. Spirillum and pallidum, Trypanoma pallidum, these are spiral organisms. Similarly, Borrelia is a spiral organism, and Helicobacter pylori is a helical organism. So these are special anatomies. Quickly to share with you, this is a culture plate where Proteus is swarming. The film of growth is the swarming. It is swarming means it is moving on the surface of the medium and producing these waves of swarming. We call it swarming. Once you see this, you say that this is Proteus mirabilis or Proteus vulgaris, which is a normal flora of the gut, but it is a pathogen. It can produce UTI, bacteremia, any pyogenic infection in a hospitalized patient. So it is an opportunistic pathogen. These are colonies, typical colonies of Staphylococcus aureus. The hemolytic colonies are the Staphylococcus aureus. The colonies which are not showing any hemolysis are the skin normal flora, which we call Staphylococcus epidermidis. Epidermidis means it is in the epidermis of the skin, it is normally living. Now, this particular pus swab, which was collected from the patient, remember I told you about the collection, selection, transportation of appropriate samples to get proper results from the lab. Unfortunately, sometimes you cannot avoid the normal flora contaminating the pathogen. Here, the normal skin flora is present along with the pathogen, which is the Staph aureus. Staph aureus has these hemolytic colonies. There is clearing around each colony, complete clearance of the blood. This particular medium is 5% sheep blood agar. We prepare agar, we put 5% sheep blood, defibrinated blood, mix it and then prepare this medium. So the blood cells are completely hemolyzed. You see the clearance around the colonies, that is the Staph aureus, which is the pathogen. The other non-hemolytic, not clearing the blood, these are the epidermidis. If the microbiologist is not properly trained, he is unable to make out this due to lack of training or knowledge or experience, he may pick all the colonies and put them into susceptibility, in which case the results will show MRSA, methicillin resistant Staphylococcus aureus. And then the doctor may start using unnecessary vancomycin and other unnecessary broad spectrum antibiotics. So it's very important for the microbiologist, well-trained microbiologist, to pick this single hemolytic colony and then do susceptibility to get actually Staph aureus. The same plate on another medium called MacConkey, you see the mixed colony types. This is how bacteria look like in a microbiology laboratory. The yellow colony, golden color colony is Staph aureus. The dark pink colony is E. coli. The deep pink colony is Enterococci. And then the pale purple colonies are Staphylococcus epidermidis. So on this particular plate, there are 4 or 5 types of bacteria. They all produce different colors, because they are all different genus, different species. They all produce different colony morphology. So a trained microbiologist who knows what these are, he will pick them and purify them, separate them, and then identify which is a pathogen, which is a normal flora, which should be reported to the doctor with antibiotics, and what antibiotics should be tested for which organism. All that is a training of the microbiology technology. This is the same which has the purple colonies, pink colonies, which is Staph epidermidis. The yellow colonies are Staphylococcus aureus, which is the pathogen. And this is a typical urine culture plate, which has mixed culture. And in urine, mixed cultures should never be reported, because these are all contaminants. They are all growing from the urethral flora or from the fecal flora, if it is a child or if it is an adult whose hygiene is wanting and the urine sample was not collected properly. And remember, urine culture should always be done with a midstream clean catch sample. There is a protocol for collection of urine sample. If that protocol is not followed, this is how urine is growing. Now, provided the tech is well trained, he will not do any susceptibility on this. he will simply report to the doctor that mixed heavy growth, contaminants are growing, please repeat the test. That is what a trained microbiologist will do. Unfortunately, if the microbiologist, which is very common in our setting, lack of microbiology training, knowledge, technical training, lack of mentorship, lack of support, this can be subjected to antibiotic susceptibility. The tech may pick some of these colonies and give susceptibility, and the doctor may use antibiotics. So this is an example of microbiology promoting unnecessary antimicrobial use and microbial resistance. This is a typical gram smear. It is showing some leukocytes, lot of necrotic debris, and gram-negative cocci in pairs. And by the way, these organisms were actually Acinetobacter. This smear is from my lab. These are gram-positive, the purple filaments. And all this is pus, very purulent sputum showing some necrotic polymorphs, leukocytes, and purple filamentous interrupted bacteria. This is typical nocardia. Nocardia are filamentous bacteria. They are normal environmental contaminants. They can be in the soil, but they can produce pneumonia in an immunocompromised host like a patient who is on steroids. So this particular patient was on corticosteroids. He had sarcoidosis and he came and got admitted. Unless this diagnosis, specifically this diagnosis went from my lab, patient was on so many antibiotics for 6 weeks, the fever was not coming down. But once we did this diagnosis, patient went on Septrin, that is the choice of treatment for nocardiosis, Septrin or trimoxazole. Patient recovered within 48 hours. It was fantastic. His fever came down. He was sitting up, drinking soup, very, very gratifying to the microbiologist. Thank you so much for your attention. I hope I have given you very, very brief glimpses into the world of microbiology. Those who are not actually familiar with the microbiology lab, my purpose is to make you interested. Take time, come and visit microbiology lab. One of the good labs, Kenyatta National Hospital, or even Aga Khan University Hospital. Take time, look at the microbes, try to understand them, because all medicos, including paramedicos, nurses, pharmacists, and laboratory technologists, clinicians, everybody should have some basic understanding of microbiology, how microbiology lab works, so that you are able to support the control of AMR in our nation, as well as support of antimicrobial stewardship by basic understanding of microbiology and how bacteria develop resistance, how antibiotics work on the bacteria. Thank you very much for your attention.
Thank you so much, Professor Revadi. You've really kept time. I appreciate you for that. And you've really merged the theoretical and the practical aspects. It only takes a guru like you to do that. We appreciate you. Before I allow Dr. Lena to say something, I would remind the participants to raise their questions on the chat box. If you have anything she has maybe reinforced, or something new that you've come across, or anything you want her to clarify, or a concept that you liked in our presentation, please, I would like to see your comments. You can even appreciate Professor for that. Dr. Linus, you can, the floor is yours. You can make your comments before we have the next key speaker. Thank you.
Good afternoon, colleagues, and it's really exciting to hear Professor Revadi on his pet topic on microbiology, and I think as the topics back to the basics, she has really brought it very clear and very understandably for us, just to be able to understand how this issue we are talking about, antimicrobial resistance, comes. One thing that Prof has been given this chance is really to thank you and to thank the participant for joining this. And I think in this forum, we're able to learn from each other. And I would really ask that even if you have cases from your sites that actually you can share on this platform will be very good, so that everyone can learn cases of how successfully maybe you're under resistant patient in terms of AMR, how you went about it, what challenges you had, or even cases that you are difficult, and at least we have experts here who can help even in addressing to that. Prof. Rebadi, I would really love if you could really comment about the biofilm formation. One of the things that we must be cognizant of is that since devolution came, I think the healthcare system has expanded, and one of the things that excites me is that, as you all know, previously patients on dialysis used to queue up for months in Nairobi, KNH, and Nairobi and Aga Khan hospitals for dialysis. But now I think we have dialysis across the country. I don't think there's any country without dialysis machine. And having this, it means that then in terms of peripheral and central line catheters, which really becomes another issue that we may be coming up with in the coming days, even after expanding this dialysis, we may be building some outbreaks that we may not be aware of. And so I would like, Prof, from the microbiology perspective, just to share with us about how the biofilm formation is contributes to AMR. Over.
Thank you, Dr. E. That's a very good question. Biofilms are formed by some bacteria, especially something like coagulase negative staphylococci, which secrete what is called slime. Slime is a mucinous material, the cell wall product, and the bacteria are regularly secreting this. All bacteria secrete it, but some bacteria are notorious. Some yeasts are also notorious, and some environmental bacteria are very good slime producers. So now this slime connects several bacteria together. And not only one type of bacteria, one genus or species, it can also connect the other bacteria which are friendly. And by the way, bacteria also produce what are called bacteriocins. Bacteriocins are antibacterial substances, like Klebsiella produces klebocin to counter E. coli. Where Klebsiella is growing, E. coli may not grow because of the klebocin. Now, pyocin is produced by Pseudomonas, and pyocin does not allow gram-positive organisms to multiply. It is an antibacterial substance to protect Pseudomonas, pyocins. So similarly, there are so many bacteriocins, but within them, some bacteria have understanding. So they produce the slime in excess on a surface, especially these catheters, even in the vascular endothelium. Then there is, supposing there is a necrosis in a deep organ, and in that space, they can produce this, organisms produce this slime, and they connect each other and they form a sheet, a layer of bacteria. Now, inside that, it is not colonies, it is communities of organisms. And these communities are highly resistant to antibacterial activity. The antibiotic which you are giving to the patient cannot penetrate this film, the biofilm, the sheet of slime. And the MICs of these organisms within the biofilm are 1,000-folds higher than the organisms which are circulating in blood or body fluids. So you pump the patient with antibiotics, but you are unable to clear the organisms which are now forming biofilm inside the endovascular epithelium because of a catheter or another prosthesis in a bone or whatever. So it becomes very, very difficult until you remove the prosthesis or the catheter. Antibiotics are unable to treat the patient. So there's a lot of research going on the biofilm to inhibit the biofilm, to have antibacterial coated catheters, and then how to counteract these communities, how to split the mucinous layer. Lot of research is going on. I think for lack of time, I stopped there, and maybe now the case presentation, we will ask Maurice now to tell us what next. Thank you.
Yes, thank you, Professor, for allowing us to continue with the next key presenter, and then we can continue taking questions on the chat box. I hope you still be with us to respond to them. Thank you, Prof. And now I would want to introduce the next speaker. She is Irene Wawero. She's an infection control nurse at Khan University Hospital. She owns a Bachelor of Science in Nursing from University of Nairobi, and has over 9 years of clinical work experience, infection control and clinical work. So I would like to introduce Irene Wawero. Welcome, and take us through the case presentation. Thank you so much, participants. As Irene is putting up the screen, I will also remind you to keep asking questions, and our keynote speakers will be happy to respond to them at the end of this. Thank you.
Good afternoon, everybody. Maurice, am I audible?
Yes.
And thank you
Yes,
for the
you're audible.
Thank you for the introduction, Maurice, and good afternoon to you all. I'm going to take you through a case presentation where we'll focus mainly on infection prevention and control aspects in hospitals, and especially among patients requiring multiple isolation precautions. Just as a brief synopsis to my presentation, we all know what has been happening in the COVID-19 era and the various airborne precautions that have been necessitated for such patients. So in the event we have patients with such requiring more isolation precautions, what do we give precedence? What are the challenges in doing so? So those will be the main focus of my presentation today. So again, this is a hypothetical situation scenario that has been modified for it to be as informative as possible for our audience today. Thank you very much. So we'll talk about Mr. FWK, who is 65 years old. He was admitted with a primary diagnosis of a critical COVID-19 pneumonia. Other diagnoses that came up as part of his management included a septic shock with an MDR cleft pneumo, a candidemia, an acute kidney injury, an upper GI bleed. He also had comorbids of the diabetes mellitus, and an acute liver injury. This patient was admitted on 7th of September and died on 10th of October. So that's approximately a month plus a few days hospital stay. So about our patient, he had received a dose of the COVID-19 vaccine on the 29th of August of this year rather. He was initially nursed in an isolation ward for 6 days. And in this case, I'd say a general isolation ward where airborne precautions were used for all the patients. Given his diagnosis, his primary diagnosis was a COVID-19 pneumonia. He was thereafter managed in a critical care unit for 27 days, where an MDR nuclear pneumo was isolated in 3 other patients. So his invasives and the invasive procedures that Mr. F underwent was that he was intubated and ventilated on the 18th of September. He had a central venous catheter. The initial one was a right internal jugular catheter that was inserted on the 18th, and removed on the 29th. Thereafter, he had a left femoral that was inserted on the 29th and was removed on the 6th. For the purpose of this discussion, we will just focus on the dates or the relevant invasives I'm going to highlight. So thereafter, therein, he also had an arterial line that was inserted on 18th of September and another one that was inserted on 29th of September. He also had a hemodialysis catheter that was a right femoral one that was inserted on the 29th of September and was removed on the 6th of October. He had a urinary catheter that was inserted on the 18th, and as per our policy, or per the policy of the institution, the patient was managed in. It was changed every 2 weekly. So the organisms that were isolated in his samples was that between first of all, as an introduction, is that between the 7th and the 27th of September, Mr. F had no organisms isolated. On the 27th of September, we had an initial organism that was isolated of a staph hemolyticus. And as Prof has told you before about the presence of this bacteria on skin. So we'll continue with the discussion, but I can say for now that this was considered a contaminant. On the 28th of September 2021, a urine, a tracheal aspirate, and a blood specimen grew a carbapenem-resistant Klebsiella pneumoniae. On the 29th of September, a blood culture also grew a Kleb pneumoniae, the same MDR, and as that, again on the 1st of October. On the 4th of October, a blood culture grew a Candida parapsilosis and an Enterococcus faecium. So use of antimicrobials, here we'll focus on the duration of use of these antimicrobials, and again, the clinical indication versus the therapeutic indication. So these were the antibiotics that
we are listening to something.
pardon me, someone might have to mute their mic. I'm not sure who that is. Maurice, maybe you can help. So again, on antimicrobial use, we will focus our discussion on the aspects of the duration of use and the clinical indication versus the therapeutic indication. So initially, Mr. F was on tazocine, which he got for 2 days. That is between the 19th and the 22nd. Of note is that Mr. F's condition deteriorated on the 18th. Please note the onset of the commencement of the tazocine. Again, he had a meronem for 3 days. That is between the 28th and the 30th of September. He had an amikacin status on 29th of September. And upon culture and sensitivity tests of the Kleb pneumo, he was begun on polymyxin and vancomycin, that is on 29th and on 30th, as well as rifaximin and micafungin for the candidemia. So given his diagnosis, he had a primary, his primary diagnosis was a critical COVID-19 pneumonia. And as is happening with the management of COVID-19 pneumonia, they are getting high dose steroids, some I don't know, dependent on the prescriber's duration of use, again, based on their own best practices. So for our patient, he got dexamethasone 6 milligrams once a day. That's on 7th of September to 8th of September. He also had a dexamethasone 8 milligrams twice daily from 8th of September to 15th of September. So having said that, I'll just go back a bit and give you a few things to note. Is that his admitting diagnosis was a critical COVID-19 pneumonia. Of note is that he had a cleft pneumo and an acute kidney injury with a candidemia and diabetes. So then he was initially nursed in an isolation ward and then managed in a critical care unit for 27 days. And in that same unit, an MDR pneumonia, Klebsiella pneumonia was isolated in 3 other patients. So he had several invasives, but and his condition is said to have changed on 18th of September when he was ventilated and intubated on that day. And on the same day, commencement of all these invasives was done. So about a few days into 2 weeks or so into the management of the patient, that is on 28th of September, that is when he grew a carbapenem-resistant Klebsiella pneumoniae in all urine, tracheal, and blood cultures. So then he had various antimicrobials, the initial tazocine, meronem, and the initial tazocine, in this case was considered to have been for the clinical indication of the patient, as was per the discretion of the consultant physician who was managing him, as well as in a therapeutic indication that was following a culture sensitivity of the cleft pneumo. In this case, it was sensitive to polymyxin and vancomycin and rifaximin, which was given on those days, and also was commenced on micafungin following the growth of candida. And again, he has had history of steroid use. So to form part of our discussion today, we look at the non-modifiable or patient factors that would have led to the outcome as it were for this patient. This is a patient who had COVID-19, who had a comorbid of diabetes mellitus and all others as mentioned in the previous slide. He's an elderly man, he's 65 years old. He had a long hospital duration, a long hospital stay, which is a month and about 3 days. And the nature of the management that Mr. F required, in this case, the need for critical care admission. So again, thereafter, I'll focus on the factors or IPC factors that were modifiable in this case. Hence, is that the practice in terms of number one, hand hygiene. What was the practice of hand hygiene intra-management of these patients in the critical care unit, which is the period which the cleft pneumo was isolated in this patient. Again, the care of invasives, you must have all heard about care bundles, in this case for a central line. So at what point did, or what bundles were not, or did not serve their purpose? Were they the insertion bundles or the maintenance bundles? And this can be depicted from the central venous catheter that grew, that was in when the organism was isolated, and the period it had been in for in that period. So again, isolation precautions in this patient. This is a patient who had COVID. So again, initially there was a no diagnosis since admission. So airborne precautions were instituted as soon as the patient was admitted. As soon as the cleft pneumo MDR was isolated, the patient then had contact precautions instituted. So again, I will focus in a bit more detail in subsequent slides regarding the practice factors, and in this case, that would have been modifiable in this case. So again, the drugs or the use of drugs in these patients, as the Prof had mentioned previously, when it comes to AMR and antimicrobial stewardship, this is a patient who, did this patient have a scenario of overuse of antibiotics? Was there overuse of steroids of these patients? Were the dosage too high? You know, just to mention but a few of the factors that would come into our mind as we would want to discuss this further. Another critical IPC factor would have been the equipment cleaning that is being used amongst these patients in this unit, seeing that we have already isolated an MDR bug in one of their patients. So are this equipment being used for one patient? Are they shared amongst patients? Did we do all that is required when it comes to contact isolation precautions? Or I don't know, was our mind majorly focused that the patient or on the airborne precautions of these patients in this critical care unit? And if I may mention, is that this critical care unit was mostly or majorly being used for COVID-19 patients. So again, back to my initial comment regarding the multiple isolation precautions that were required. So having said that, this is a bit of a busy slide, but nonetheless, allow me to say that a summary of a few of the IPC aspects that we can learn in this case is, again, the multiple isolation precautions. We note that the airborne precautions were instituted initially due to COVID-19, and contact precautions again were instituted when the MDR bug was isolated. However, did the airborne precautions take precedence over the contact ones? Were they ignored in between patients who required both airborne and contact precautions? In this case, as we had mentioned, patients with a similar diagnosis and in the same unit also isolated the same bug. So neutropenic precautions. So this is a patient who was on very high steroid dose therapy. This is a patient who we would have considered for neutropenic precautions. And that said, then how do we combine all these precautions when providing care to one patient in the midst of other patients who have similar diagnoses in a critical care unit, who, as we all know, required mostly a gown and an N95 mask. Another aspect of IPC would have is the choice of the CV site, the central venous catheter site. Initially, it was in the left jugular vein. Subsequently, it was in the right femoral, but again, saying that the patient also had a hemodialysis catheters that needed to be changed like that. So we would say that the initial choice of the jugular was preferable. And again, that doesn't seem to be much of a factor. So then let's focus on the care of the central line, the central line care in this case. So again, during insertion of this central line, were all measures taken, in this case, the insertion bundles, all the effective practices that have been shown that when performed together will lead to better outcomes. Were they done accordingly? As per our case, seeing that we grew the bag, we'll check later, a few days into the central line being there, then we cannot quite allude it to the insertion, but rather the maintenance bundles of maintenance of a central line for this patient, who then also had COVID-19 and in this unit that had all these things going on. So let's talk about equipment. This is a unit that is critical care that was, that needed auscultations to happen for patients as such who are ventilated. So an equipment that can be a culprit in this case can be a stethoscope that can be used from a patient in room A to room B to room C. And in that case, if that's happening in the crisis of COVID-19 and all, were we following the recommended precaution guidelines? So again, when it comes to geographical considerations of this thing, this bag was sort of isolated or confined into critical care units, which is quite expected in such bags. It is known that these MDR bags are mostly isolated in critical care units. So basically, this is in keeping with what is known there already, that the bag circulated in the critical care units in patients with similar diagnosis, and hence again the isolation principles above. Again, the clinical team that managed these patients, did the patients who got the MDRO, were they managed by the same clinical team? In this scenario, all the patients who had the MDRO were managed by the same clinical team, which is just to say, go back and ask ourselves, did we airborne precautions notwithstanding, were the contact precautions exercised in between these patients when it comes to their CVC maintenance, care, in terms of equipment use amongst these patients, in terms also of the geographical location? It was circulating around a unit, it was confined in a unit. So that means whatever PPE we don't or didn't, for that matter, was confined to that unit, because there's no way, I don't know, people leave an entire unit with PPE. So, which is to say, again, contact precautions were the key thing that we were looking at in today's discussion. So, a brief summary again is that hospitals breed many infections, and how good we are to detect them makes the difference. Hence, Prof. Rebathi's laboratory, the role of infection control nurses and infectious diseases team in terms of antimicrobial stewardship, in terms of diagnosis, in terms of confining and management of the organisms of the infection. And thank you very much. And I'd summarize and say the germs are very small, but are still very scary, and infection prevention works. Thank you very much.
Thank you. That's wonderful, Irene Waweru, for reminding us the basics of IPC in terms of practically how we need to enforce them in the facilities, and asking ourselves, where do we go wrong in terms of IPC? So we appreciate your comments regarding Irene and Irene presentation, sorry, as I start off with the comments from Professor Rebathi. Are you ready, Prof?
Yes, Morris.
Okay. We have one question here that I'll read, and then I can maybe break it down. First of all, I would like to let you know we have so many comments appreciating you and the knowledge you have imparted on the many of us. Starting with myself.
I'm very thankful. I'm very thankful for the appreciation.
Thank you. I think I also led I read one of the guidelines that was saying we collect a pass swab by putting a swab deep into the abscess. But today you've really taught me something different, that it matters on the state of the hood, and that matches with the question that Charles Wawero is asking. That Prof, thank you so much for the presentation. You spoke of a mixed culture results from contaminants being reported to clinicians by some lab technicians. How is that so? So should there not be checks and balances of what is reported? In other words, the technician or technology at the bench will do the test, and maybe someone senior will do reporting after checks. So this question, Prof, really puts the issue in context, is the issue of sample collection, sample management, and quality assurance is a key issue in lab. I believe Charles would want to know what are the checks and balances to ensure accuracy of the results, as well
Can
as—
I quickly answer that? I have
Yes.
already answered that question to him on the chat, but I
Okay.
didn't address everybody. You see, we don't have clinical microbiologists in our environment. That is number one we are suffering from. We
Yes.
have microbiologists who are trained on the process, that is how to inoculate, how to identify those things. We have done a lot of training, how to pick bugs, how to identify. Then a lot of automated systems are there. If somebody can generate a pure culture, put it into the system, it will do the identification and antibiotic susceptibility also. The process training we have done a lot, but what we don't have in our environment is the interpretation of microbiology with the clinical angle. How to engage the clinician, how to report the bug which is clinically significant. Like the urine culture example I have shown, it's a mixed culture, but it is only a clinical microbiologist who has that training, which is uropathogen, which is not uropathogen. It's not that simple. We cannot oversimplify things. If you take Enterococcus, it's normal flora in urine. It's a fecal organism. Always it contaminates urines. It will appear in the culture plates. But the techs are trained to read the plate, to do ID and susceptibility. So they pick all enterococci, they report the susceptibility, and then which drug to test on which bug. This knowledge is very, very poor in our environment. This is again because lack of clinical training. So we need to train large number of clinical microbiologists. Unfortunately, our ClinPath program in Aga Khan has trained quite a good number of people with a good microbiology laboratory. To train them, you need a good clinical lab also. That is also wanting. We don't have so many labs which have capacity to train people. So it's a mammoth task. Right now, the microbiology, if you have half-baked microbiology, a tech is operating alone, there is no mentorship, nobody to hold his hand, he is releasing sensitivity results, and we are promoting AMR. That is my fear. It's happening all the time. So this forum, I think due to limitation of time, I may not elaborate on it, but that is my answer. Thank you.
Okay, thank you, Prof, for that question, that response. Sorry. As we invite more questions, and before I turn to Irene, Prof, you've mentioned this is your mixed culture results and originate from the sample collection, and you've mentioned on aspect of pass swab collection. Would you want to mention on some other samples we take commonly, like sputum, like urine? Where is the contamination coming in?
Excellent, thank you. Sputum comes with the normal flora. Respiratory tract is full of normal flora. So unless somebody is instructed properly, supervised, all people don't know. Simply you give a container, go and collect some sputum and give it. That's all is told to the patient. You have to instruct the patient. If early morning before breakfast, please collect the sputum. Cough deeply, do a good cough, bring out the deeper collection. Don't spit into the container. These things have to be very clearly told. Now, if it is a spot collection, patient has come to the lab, make him rinse his mouth with fresh water. or distilled water. Rinse your mouth, remove all food particles, then collect the sputum. It has to be deeper collection. So this is a training, and it is paramedics who can do it, and doctors also have responsibility. Urine in babies, in children, in females is very, very challenging. Patient needs elaborate instructions how to collect urine. It's a whole protocol. Then comes to the blood collection. Decontamination of skin is very, very specific training. Unless it is done, all blood cultures are growing, coagulase negative staph. All techs are releasing antibiotic susceptibility because they don't know which is normal flora, which is pathogen. And some labs have thrown away Staph aureus from blood culture because they were told that staphylococci are contaminants. Some mentor came quickly in the lab, did a lecture, told them staphylococci are contaminants. So the techs are throwing Staph aureus, which is a top class pathogen in blood culture, very dangerous. They have been throwing them. Then yeasts are contaminants in blood culture, somebody told them. So KNH lab has been throwing all the yeasts, which is a very, very killer pathogen in the blood culture in ICU patients. But the lab has been discarding them because they did not have the clinical input. So all samples have protocol, and all samples grow normal flora, but clinical training, clinical correlation is very, very important, especially urine culture. Clinical correlation is very important to interpret every plate. You need minimum information. It's a lot of training, takes at least 1 to 2 years. It can't be done in a mentorship of 1 week or 1 hour or 1 hour lecture. It is not possible, unfortunately. Thank you.
Thank you so much for the elaborate responses, Professor. We have a lot of people concurring with you. Jane Gakash's excellent presentation. Kindly ensure that are training to avoid wrong diagnosis. Everybody is agreeing with you, Prof, on that. Now coming to Irene Wawero, we also have a lot of comments. Appreciate your good presentation. There is one particular question from Nelius Makadu. This is excellent presentation. Kindly confirm whether we need to change invasives routinely or as per assessment. Irene Wawero, are you able to comment on that question?
Yes, and thank you very much for the question. I think it's Nelius, you said? Yes, Nelius. So yes, this is very dependent on your institution's guidelines, number one, but best practice definitely shows that some invasives do require to be routinely changed. You will have them have explicit recommendations for some. For example, how often should you change urine catheter? For most, they say every 2 weeks. How often should you change a central venous catheter? Most of them say 2 weeks. That's with the latest infusion guidelines and things like that. But besides the invasives, small things as your infusion sets, the administration sets as well, those do require, and again, some institutions to specify on how often they require to be changed. So let's not only focus on the invasives themselves, all other things that come with the invasives, including the catheter itself, the infusion set or the administration set, the arterial line. Are you giving fluids? Are you giving blood? Are you giving TPNs, total parenteral nutritions, other nutritions, then lipid-based or not? So to answer your question, yes, we do need to change invasives routinely, and many guidelines, whether you're using the infusion ones and things as those infusion nurses guidelines, then we'll give you that, but it's very dependent on our formulated as per policy guidelines in every institution. Thank you.
Okay, thank you, Irene, for the response. And now I would like to invite Lydia. She would like to share with us how we can provide feedback regarding this session before we come to an end. Lydia.
Thank you very much, Maurice. I have posted a link on the chat box. If you could kindly click on it and give us a feedback of what you thought of today's session. Also, to just let you know that IPNET Kenya is organizing its 9th conference, which will be on 23rd to 25th March in Nakuru. We have opened up for abstract submission. So if you would like to let us know what you're doing in your facilities, this is a good opportunity to do that. The deadline is 31st January. I think I have all your emails, and you're in one WhatsApp group or another, so I'll be able to share the poster with you as regards what the theme is for next year's conference, as well as what the sub-themes are. Then also next session will be on 6th of January. The topic will be standard precautions on hand hygiene and hand hygiene audits. And as we had been told earlier, it is good if we share some of the experiences we have in the hospital. And I'd like to ask if any one of us would be able to volunteer to give us a case presentation from your facility. Kindly just put up your hand or put it in the chat box that you are ready and willing to give the case presentation on 6th of January.
Has anyone responded, Lydia?
No, not yet, Prof.
Dr. Zahir, are you online?
Yes, Prof, I'm online.
Yeah, if you want to present from Coast General, you are most welcome, please. If you can motivate your team to prepare a case and your team can present, we'll be very happy.
We'll discuss my team, Prof, and then get back to Lydia.
Thank you, Dr. Ari.
Thanks. I'm putting my number on the chat box so that in case you need to reach out to me, then that would be okay.
Okay, thank you, Lydia. I want to appreciate the participants for the active interaction we've had. We've learned, we've asked questions, comments. We take note of them. We appreciate your time and finding time to actually listen for and participate in this kind of discussion. Let's continue with the discussion in our facilities, in our workplaces, and improve IPC in our work areas. For our key speakers, we are honored to have you, and thank you so much for preparing for this and commitment. It takes a lot to come and present in this kind of a forum. We really appreciate, we've seen the comments from the participants, they're happy and they've learned a lot from you. Thank you so much, and wish you all the blessings. For my co-host team, I also want to thank you, and unless you have something I've missed you want to highlight, I would want to call it for a close. We meet again in next month. Lydia, Nicholas, anything that
Thank
you've
you,
missed?
everyone. Everyone have a nice afternoon. Happy reading, happy learning. And also, I think we should greet Merry Christmas and Happy New Year, because our next session is in January.
Yes, sure, Prof.
Merry Christmas and Happy New Year to all.
Merry Christmas.
Merry Christmas to you.
Merry Christmas.
Merry Christmas to you.
Merry Christmas.
Merry
Happy New
Christmas.
Year.
Merry
Happy
Christmas.
New
and
Year.
Happy New Year.
Merry Christmas. Happy New Year.
Merry Christmas.
God bless you all.
Be blessed.
Merry Christmas.
Merry Christmas.
Hi, Romina, we can end the session.
Sure, Lydia. Thank you.
Thanks,
Thank
thanks,
you.
Romina. Thanks, Maurice.
Thank you.
Thanks, everyone. Bye.
Bye-bye.
Bye. Bye.